Hello, welcome back to my channel. Today we are going to discuss about gel electrophoresis. So we know that electrophoresis is the migration of charged particles or molecules in a medium under the influence of an applied electric field. Electrophoresis is broadly classified into free electrophoresis or zone electrophoresis. In the case of free electrophoresis or electrophoresis without stabilizing media, here, the separation of charged particles takes place without a stabilizing media or supporting media. But in the case of zone electrophoresis, there is a stabilizing media like agar, starch or polyacrylamide is mainly used. So, in the case of this is separation. In the case of gel electrophoresis, this separation is brought about through molecular sieving technique based on the molecular size of the substances. Here the gel material acts as a molecular sieve. Gel is a colloid in a solid form that's contain about 99% water. It is important that the supporting media is electrically neutral. There are different types of gels which have been used are agar, agarose gel, starch, Cifadex, polyacrylamide gel, etc. Here the porous gel acts as a sieve by retarding or in some cases by completely obstructing the movement of macromolecules while allowing smaller molecules to migrate freely. Agar gel is used for the separation of different types of protein mixtures as well as nucleic acids. Polyacrylamide is most suitable for the separation of nucleic acid. It is also frequently used in separating proteins, peptides, amino acids from microgram quantities of mixed samples. Then what are the classification of gel electrophoresis? First one is agarose gel. Agarose gel electrophoresis is a method of gel electrophoresis used in biochemistry and molecular biology to separate and analyze DNA or RNA molecules by size. It is mainly used to determine the size of DNA, checking the purity of isolated DNA. Agar is a mixture of polysaccharides extracted from seaweeds. Agarose is highly purified uncharged polysaccharide that derived from agar. Agarose is a linear polymer of D-galactose and 3,6-anhydrol-L-galactose. Agarose dissolves when added to boiling liquid. It remains in a liquid state until temperature is lowered to about 40 degrees Celsius, at which point it gels. The gelling property are attributed to both intra and intermolecular hydrogen bonding. Agarose gels are fragile. Agarose is used in concentration as 1% and 3%. Spore size is controlled by adjusting the percentage of agarose used. Large pore sizes are formed with low concentration and vice versa. Agarose gel have larger pore sizes compared to dextran and polyacrylamide gels. This makes it useful for the analysis or separation of large globular proteins or long linear molecules such as DNA. This is the structure of agarose. Then the principle behind this agarose gel electrophoresis is biomolecules like DNA or RNA are separated by applying an electric field to move the negatively charged molecules through an agarose matrix towards the anode, and the biomolecules are separated by size in the agarose gel matrix. The largest molecules will have the most difficulty in passing through the gel pores, whereas the smallest molecules will move faster. After the samples of DNA are applied to the wells of agarose gel along with the tracking dye, then the power is turned on after filling the buffer in the tank to allow current to pass. The tracking dye and DNA samples are negatively charged and they migrate towards the positive electrodes. Move. The commonly used buffers are either TB or TAE buffer. The buffer used helps in deliver the electric current through the gel. TB buffer is a tris boric acid EDTA and TAE is made with the tris acetic acid EDTA. A gel casting tray is used which is made of UV transparent plastic. The open ends of the trays are closed with a tape while the gel is being cast, then removed prior to the electrophoresis. This is gel casting tray and procedure steps. Loading the samples, etc. A comb is placed in the liquid agarose after it has been poured. Removing the comb from the hardened gel produces a series of wells used to load DNA fragments. A tracking dye such as bromophenol blue is also included in the sample. This makes it easier to see the sample that is being loaded and also acts as a marker for the electrophoresis front. Next is gel staining. The DNA in the gel needs to be stained and visualized. The reagent most widely used is the fluorescent dye like a ethidium bromide, ETBr. That emits orange light after binding to DNA fragments. The gel will be viewed under UV light. Ethidium bromide is a cyclic planar molecule that binds between stacked base pairs of DNA. Then the voltage applied. Higher the voltage, the more quickly the gel runs. But if the voltage is too high, the gel melts. The best separation will apply voltage at not more than 5 volt per centimeter for gel length. Determine the size of DNA fragment. Since agarose gels separate DNA according to size, the size of DNA fragment may be determined from its electrophoretic mobility by running a number of standard DNA markers of known sizes on same gel. Note that higher the voltage, the more quickly the gel runs. Gel concentration must be chosen to suit the size range of the molecules to be separate. This is the method of gel electrophoresis. First we have to prepare agarose gel, melt, cool and add ethidium bromide, mix thoroughly, then pour the mixture into a gel casting tray which is made of UV transparent plastic with a comb and allow to solidify. Add running buffer, load samples and marker, run gel at a constant voltage until bond separation occurs. View the DNA on UV light box and show results. This is a generalized diagram of a gel electrophoresis. You can use it for agarose or other gel also. It's a generalized diagram. What are the advantages of agarose gel electrophoresis? It is easy to prepare and small concentration of agar is required. Resolution is superior to that of filter paper. Large quantities of proteins can be separated and recovered. Absorption of negatively charged protein molecules is negligible. It absorbs proteins relatively less when compared to other mediums. Sharp zones are obtained due to less absorption. Recovery of protein is good and it's a good method for preparative purposes. One of the disadvantages of this agarose gel electrophoresis is electroosmosis is very high. Next we are discussing about a starch gel electrophoresis. The starch gels are prepared from hydrolyzed potato starch which is dissolved by heating and pouring to a thickness of 5 to 10 mm. The pour size can be adjusted by Starch concentration of the solution, like in the case of agarose gel electrophoresis. A suspension of granular starch should be boiled in a buffer to give a clear colloidal suspension. The suspension on cooling set as a semi-solid gel due to linkage of branched chains of amylopectin. In order to avoid swelling and shrinking, petroleum jelly can be used. Partially hydrolyzed potato starch makes for another non-toxic medium for the protein electrophoresis. The gels are slightly more opaque than acrylamide or agarose gel electrophoresis. In order to avoid swelling and shrinking of this starch gel electrophoresis, petroleum jelly can be added. Non-denatured proteins can be separated according to the charge and size. They are visualized using naphthol black or amido black staining. DNA is cut into smaller fragments. Loading dye is used to indicate fragments of DNA. The negative DNA molecules are attracted to the positive electrode. The smallest fragments move greatest distance. Then what are the advantages of this starch gel electrophoresis? Which includes high resolving power and sharp zones are obtained. The components resolved can be recovered in reasonable yield, especially proteins. Can be used for analytical as well as preparative electrophoresis. Next is the disadvantages of this starch gel electrophoresis. Like agarose gel, here also electroosmotic effect is very high. Also the variation in pore size from batch to batch. Next is polyacrylamide gel electrophoresis or PAGE. Polyacrylamide gels can be used for the separation and analysis of proteins and relatively small nucleic acid molecules. DNA monomer units has one phosphate moiety that carries the negative charge. When an appropriate denaturing agent such as urea is added to DNA samples and the gel is heated, the shape of varying length linear DNA molecules become identical. As a consequence, denatured molecules will be separated exclusively based on their size. There are several PAGE methods: native PAGE, SDS PAGE, isoelectric focusing, one-dimensional and two-dimensional PAGEs are available, and that can be applied mostly for the separation of proteins based on distinct molecular properties. At a given pH, different proteins carry different amounts of electric charge, different shape and sizes. In the presence of suitable initiator and catalyst compounds, acrylamide can readily polymerize in a radical process. So we know that acrylamide is harmful by inhalation or direct skin contact. This reaction would lead to very long polyacrylamide chains yielding a highly viscous liquid instead of a gel. These long chains need to be cross-linked to form three-dimensional network. This is achieved by mixing NN-methylene bisacrylamide into the acrylamide solution. This NN-bisacrylamide is composed of two acrylamide molecules covalently interconnected by a methylene moiety. During polymerization reaction, the acrylamide groups of NN-methylene bisacrylamide molecules become incorporated in the long polyacrylamide chains. Crosslinks are formed between the polyacrylamide chains leading to a gel. In this diagram, we can see that this acrylamide combined with NN-methylene bisacrylamide leads to long chains of polyacrylamide gel or polymerized into polyacrylamide gel. The three-dimensional molecular network comes into being by radical polymerization of acrylamide monomers and cross-linking N-methylene bisacrylamide components. Without any modification, polyacrylamide electrophoresis separates macromolecular ions based on a combination of charge, size and shape.